Okay, so this is the sheet that I want to talk a minute about. I added just a few things in there that was not on the last lab report, like the variables and so on. So I want to just talk a little bit about that. So in the title, notice that I added some specific questions that I'd like you to answer in the background information. These are things that a person from the street would, if they're reading your lab report, would maybe need to have some background information to understand what in the world your lab was about. So make sure that you answer those questions as your part of your background information there. Purpose and objective of the lab, the variables, so that's new. The variables, independent, dependent, variable. If you are doing this and you have trouble identifying the variables, please come in and see me and ask for help before Friday. All right, and I can help you to identify those. All right, and control an experimental group slash, and I put in parentheses there, groups, because in a lab you can have multiple experimental groups. And we did have multiple experimental groups in this lab. And then the experimental controls or constants, those are things that you want to stay the same between all of your groups. All right, so again, if you have any questions on that, come and see me. And then hypothesis, the rest of it, material, procedures, all of that is the same as before. For your data and observations, the title for a graph, you need to you can use the graph that you made on Friday in this class with the graph paper. Just make sure that you have those things on there, like the title, label them with including units and so on. The data table, though, that you made to record your data needs to be put into a into your lab report. So it needs to be embedded in there, typed in there, and so on. And so if you have trouble with that, if you're not sure about how to make a data table, you can come see me and I can help you with that. It's not too hard, but it needs to be embedded in there. All right, and then the conclusion. Read through the bullets there about what makes a good conclusion. Your conclusion should answer the question. Remember, you picked one of two questions to answer in your lab. Your conclusion should actually answer that question and give experimental data. So it says provide evidence for your claim. That means that you can, if you answer the question, you need to provide evidence from your data and put that into your conclusion. All right. For the analysis part, the analysis is if you pull out the initial reading about the lab, there was a stapled packet that we read about the lab. Pull that out. Looks like this packet here. The last page on this packet has 6 questions. Those are the 6 questions I want you to answer. Okay. In the analysis questions. And then a bibliography, this is only if you use outside sources. If you'll use anything that we did or talked about, you don't have to cite that packet that I just pulled out and so on. So unless, if you use everything from our class, then you wouldn't have to cite anything. But if you're using outside resources, then, all right. No, you don't have to include it if you didn't. So I'll just make that assumption. Okay? All right, and then also a couple of other things. For your hour, I think I forgot last Friday to pass back your last labs. I had them and I forgot for you guys. So I'm going to pass those back so you can see how you did on that so you don't make the same mistakes again. All right, and so I'll pass that back. Couple of things when you're doing your report, I would like all the different sections, like I should be able to look at it and say, okay, boom, this is the background information right here. So put a title, like, maybe bold it so that here's the background section, here's the procedure section, and so on. So make it, maybe leave a space between it so that you can, making it visibly pleasing. Some people did a heading and they would just write, title, and then put the title, and then all same, it all ran together, then didn't skip a line and wrote, you know, hypothesis and so on. So it all like kind of was like one big. It looked like one big paragraph. It makes it. It's okay, but it doesn't. It makes it a little bit harder to grade where everything separates and so on. And so just separate out the parts with that. And then another thing is with your lab group. That your work should be your own. I've had, in the last lab, I had people have very, like word for word, like answers to the questions. The analysis questions at the end was word for word. You know, no two people, you can work together and arrive at the same answer, but 4 people, if you sit and ask a question and have them write down the answer, the likelihood that they're going to say it exactly the same way with the exact same mistakes is not likely. And so therefore, you can work together to come up with the answer, but then put it in your own words. Okay, so and that doesn't mean like changing one word. All right, it's very obvious. All right, so don't do that. Okay, again, if you did it before. Okay. All right, so that's my thing with that. So in just a minute, I'll pass that back to you. Before I go on and we get on with the photosynthesis stuff, I want to talk to you about the whole Google Classroom thing. Did you guys get my? I sent a remind text yesterday after school, but it's no big deal if you didn't get it or didn't do it. But let me just explain here what I'm going to start doing. I'm going to put everything up, but it automatically turned off again. Oh, no, it didn't. There we go. All right, so I sent it a text to check your email. So yesterday, I sent the whole class an email with the code for this to join the class. If you did not get that email, did anybody not get the email or need the code? I can show you. Oh, can you show the code? The code here. I'll read it out loud, but I did email it to you because it is case sensitive, but this is all lowercase. It's, for our class, it's 86DN5W. And so I'd like you to join. It's the second hour AP biology. What I'm going to start doing is using this as kind of my calendar of what we've done every day. And so before I leave during the day, I'll write what we did. So I just did this yesterday, where I just wrote I'm using this announcement feature to write what we do every day. This way that if people who are absent for whatever reason can see what we did, and I'll put. I didn't do it yesterday because I was just started it yesterday, but this is the announcement feature. There's also an assignment feature, and so I can write to you an assignment with a due date. So I can click here and do assignment. So create an assignment here. I just did an announcement. So this is an announcement of what we did. I'm also going to scan in all of the stuff that you would need to do what we did in class that day. So yesterday, I included and uploaded the PowerPoint notes that you guys picked up yesterday. So that way, if you are absent, you can click on the handouts, all right, click on it and print it out and so on. Yes?
Can you scroll up so I can...
Scroll up?
Yeah, because I can't see the code right now.
Oh, that's because I switched screens.
Oh.
The code is on the other screen. Here, I'll go this way. Well, let me go to students. There we go. Here it is. It's 86DN5W. Okay, so you can print off the notes or whatever, like the handouts that you got today for today's. I will put those handouts as well, so therefore everything will be there. And then I also uploaded the video here, so you can click on that and it'll go right to the video. The videos, I'm going to start putting it as an assignment, so it'll be as an assignment. And you will get, to my understanding, for students, you get a announcement, so a little banner comes up on your phone when you do this, kind of like if a game is updated or so on, where it'll say you have a new announcement. Also, I think an email is sent here. now.
You can turn that on and off. But
You
yeah,
can turn that on and off. So if you, how do you, as a student,
I don't
is-
remember. I just know I had it turned off
okay,
for my
all right.
class.
So you probably, as a student, can adjust your settings, all right, in terms of that, okay? So, so that's the plan. So that way everything is all in one spot. Because I did have a website, I was putting stuff on a calendar, and then, you know, this is, everything's all in one spot, and I think people will use this more. All right, so that's the plan from here on out. So you can access the video from YouTube, or you can access it from there. Okay, all right? All right. Any questions on that? Oh, and you can put announcements and put it under a topic. So I did this as Chapter 10, Photosynthesis. That way over here, you can sort by topics. This, I think, will become important maybe for studying for our final exam or the AP test. If you want to study or relook at certain things, you can go, okay, I didn't do very well in photosynthesis. You can go and find the photosynthesis topic and click on it, and it'll give you all the stuff just pertaining to photosynthesis. All right, so that's an option as well. Okay? Alrighty, so that is Google Classroom. Okay, so what I'm going to have us look at now is the photosynthesis stuff. So you picked up the I can statements, which is like a checklist of what you need to know for photosynthesis. And we, from last night's video, you should be able to do all of number 1. All right. So those are things that you should be able to do. Tonight's video is going to be about number 2. All right. So, so what I'd like to do now is, remember the I can statements are not due, they're just to help you to guide your studying to make sure that you can do those things. If you take a look at this single sheet here, this one here with it says interactive question 10.1 and interactive question 10.2. What I'd like you to do is look at those 2 questions and without looking at anything, see if you can fill in the blanks. So this I'm modeling, this is part of what you should do for studying, all right, is to see if you can produce off the top of your head. So outer and inner membrane there. Then I skipped over B. B is the stack of these little discs here. The whole stack is called what, Olivia? The grana. The granum is singular, grana is plural, so this is one stack. What are they a stack of? That's what E is looking at. E is looking at one of those little guys here. One of those that are stacked up is what? Ellie?
Thylakoid.
A thylakoid. So then, if you look at what D is pointing to, so E has that little bracket for one of these guys. D, what do you think D is pointing to? Huh?
The thylakoid space.
Yeah, it's hard to see where it's pointing to. The thylakoid space inside of there. Anybody put thylakoid membrane? No? Okay. Could be the thylakoid space. That's what it actually is pointing to. But the membrane is around the outside too. I was just wondering if anybody put that. The thylakoid membrane is, you know, the outside. If this is the thylakoid, we looked at the membrane here and enlarged it. It's a lipid bilayer, just like the cell membrane, and with proteins embedded in it. What's the purpose of the membrane of the thylakoid? What's the role of that? Does anybody know what it's embedded in those membranes? Do you remember from the notes? The membrane of the thylakoid, that's important, really important for photosynthesis. It stores the pigments. So the green pigment in plants is called what? Chlorophyll, which means it reflects green light, which is why it appears green. So, and where those pigments are in these thylakoid membranes. And then lastly, F here is pointing to the space inside the chloroplast. This space is filled with a fluid, and that fluid has a special name. That special name is what is found? The stroma. All right, and so that is our chloroplast. Now, when we look at just the general overview of a chloroplast, that's this other picture here. So here's the chloroplast. Here's your granum that has the thylakoid stacked up on top of each other. And the rest of the space, they only did one of those for simplistic sake in this picture. And the rest of it is that fluid called the stroma. And so these letters C and H, these two are referring to the two main phases of photosynthesis. What's the first phase of photosynthesis called? Joel? The what?
The light reaction.
The light reaction. So these guys are the light reactions. Implying that it uses light. So what do you think is represents light here? The little squiggly line here. Usually our book uses like the little jagged lines like that for a form of energy. So in this case, the form of energy that is coming in is light. So this is like the light that's shining on the plants. So the light reactions are first. The second part of photosynthesis goes around and around. Sam? Is the Kelvin cycle. This is named after the person who discovered the Kelvin cycle. His name was Melvin Kelvin. All right. So he came up with the Kelvin cycle. Now, the light reactions here, besides light, the light energy is used in the light reactions, but remember photosynthesis, the overall reaction, there are two things that go into it besides light, and that is, what gas goes into photosynthesis? CO2, and what's the other thing that goes into photosynthesis? H2O, right? Which one of those, we already have light, is it CO2 or H2O that is used in the light reaction? Hannah? Water. So we have H2O here. And remember that the energy from the light is going to split water and split it into its respective elements. And so we have hydrogen and oxygen here. And so when it splits it, part of the hydrogen, remember, carries electrons. Each hydrogen has one electron. So the electrons are important for the rest of photosynthesis. So what is gotten rid of? What's the part of water that the plant doesn't need? The oxygen part. So therefore the oxygen part leaves the plant, and this goes into our atmosphere. And this is the oxygen that we breathe in to use for what? What do we use oxygen for? Cellular respiration to make ATP. All right, so then the hydrogen part. Where do the electrons and hydrogen go? What are they going to become part of? We look at here what goes into the light reactions, NADP plus and ADP in an inorganic phosphate. Which one of these is going to carry the electrons and the hydrogen, Ellie?
The NADP plus.
The NADP plus, and when it reacts with these two hydrogens, it becomes NADPH. And just like NADH in cellular respiration, this is carrying two electrons. Then, last thing that's made, putting these two together, ADP and a phosphate, is a what kind of reaction? Endergonic reaction. It needs energy. The energy comes from the light. The light helps to put the phosphate back onto ADP to make ATP. And so the light reactions, the goal of the light reactions is to get these two molecules that now are used for the Kelvin cycle, because this first step, remember the whole goal of photosynthesis is to make what molecule that we haven't made yet? Sugar, right? That's what's going to come out here is the sugar molecule. So this is going to be sugar, and the basic formula of the sugar we wrote in our notes is CH2O, but glucose as an example of that, follows that same formula. Eventually we get glucose. And so, so that's what we're going to make. And so the ATP and the NADPH are used in this Kelvin cycle because what molecule is the second reactant that we haven't used yet? CO2, and that's the C and the O as part of the sugar. H's from the NADPH are going to be the H part of the sugar. And so CO2 goes through a series of reactions which require some energy in those electrons to make your sugar molecule, and then regenerates that original molecule so then the next CO2 can come in, and so on and so forth, and you get this cycle. And so this is the overall purpose, to get that sugar molecule at the end. And so what I just did with putting, not only putting what comes in, but kind of explaining it, was what you needed for me to be able to do that. All right, because tonight's video we're going to look at more in depth about what happens in the light reactions and so on. So we're going to look more in depth. That helps to understand if you have the big picture down. All right, so what I'd like you to do next is look at the other side of this. The other side is we're going to talk a little bit about which wavelengths of light, as it says at the top, are most effective in driving photosynthesis. So what I did is I put two different graphs on here. The instructions for the graph, each graph are the same. So it says the following graph, this is called an absorption spectra. Look at the graph and summarize below what you learned from reading the graph. So one thing I noticed after I made the copies is the x-axis, I forgot to label what these numbers are. These guys, these numbers are wavelengths of light. And so remember, white light is all the different wavelengths or colors of light, the visible spectrum from violet to red, so it's the whole Roy G. Biv thing. So that's your wavelengths of light. And it says on the y-axis, it says absorption of light by chloroplast pigments. So these right here, these three different lines, chlorophyll A, and chlorophyll B, and carotenoids, these are all, guys, pigments found in a plant, different types of pigments. And so what I want you to do is to look at that and write what this graph is telling you. And then I want you to do the same thing with the next graph. The next graph, the y-axis is different here. It's the rate of photosynthesis. The x-axis is the same. So wavelengths of visible light. And then I want you to write a summary from what that is telling you. And then looking at both of these together, I want you to, at the end, to draw up a conclusion about what conclusion can you draw from the graphs about the initial question. The initial question is what wavelengths of light are most effective. So, so what I'm doing here is giving you practice interpreting graphs. This is something that you will also need to be able to do on the AP test. They may have graphs that you have to look at and interpret information from it. So this is a little bit of practice. Okay. So I'm going to give you some time to do that right now. And if you're confused about it, you can talk amongst yourselves to work together on it as well. So when we look at this first graph with the wavelengths of light, somebody volunteer and tell us what you can learn about what's this graph telling you? I'll volunteer at once. Megan?
Saying that chloroplasts absorb high amounts of light with wavelengths in red and blue-violet regions.
Yeah, like if you, now obviously I can't make copies in color, but let me show you the picture that I made the copy of in terms of here so we can see the different colors of light. So we have these different pigments, and so the pigments, as Megan said, have high absorbance in the violet and blue range and in the red orangish range here. And how we know that is because of the peaks here. Notice that each different type of pigment, chlorophyll A, chlorophyll B, and the carotenoids, have peaks at slightly different wavelengths there. But if all of these pigments are found in the plant, then they will absorb all of these wavelengths of light in that range. So from like what, 30-ish to 500 there in that particular area. And then we see it all go down here. There's no peaks in the green and the yellow, and then we have the peaks again in the longer wavelengths. What does that tell you about the green and yellow? What's happening to them? If there's no peaks, that means there's very little absorption, which means what is happening to that light then? It is being reflected. So what happens is the plants that appear green, they may have this orange pigment, carotenoids, but the plant still appears green. That's because they have way more chlorophyll pigments than they do other colored pigments. That's actually what we just saw and witnessed this fall, you know, the leaves changing colors. And so the reason why they change colors and why they appear a different color in the fall is during the summer, they have a bunch of chlorophyll. They have way more chlorophyll than other pigments, like the orange or red pigments or yellow pigments. They have way more chlorophyll. Then what happens in the fall is the temperature drops. The dropping of the temperature triggers the plant to destroy the chlorophyll. So the chlorophyll actually gets destroyed. The other pigments do not. So that leaves the orange pigments now. You can actually see the orange when the light shines on it. The orange pigments reflect orange light, and the leaves appear orange, and they don't appear green anymore. That's why we had a really warm fall, and the colors were pushed back. Did you guys hear about that on the news? They kept talking about the colors were a week or two behind, like the changing of the colors. That's because it took longer to dip the temperature to dip low enough to trigger the chlorophyll to be broken down and so on, and the colors to change. All right, so these pigments that we see in our leaves right now, the reds and the oranges and so on, were always there in the plant. They were masked by the chlorophyll. Okay, so this shows you what wavelengths are actually absorbed. Next picture. Next picture, the y-axis is different. Rate of photosynthesis. So what can we conclude here from this graph about, obviously it's going to be about the rate of photosynthesis because that's what was being measured, that's on the y-axis. So what can we conclude here? Serena, what do you think?
I said that from like 400 to like 450-ish, it's increasing, but then it decreases until like 600.
Okay, all right. And then picks back up here, right? All right, with peaking at what, maybe 680 or something like that. All right, so therefore, we can see that the rate of photosynthesis is higher in these peaks, and therefore these wavelengths between 400 and 500 here we have a peak, and what, like the peak here, 650 to 700, we get a peak. So therefore we have higher rates of photosynthesis. Notice that they're measuring it by how much oxygen is released. The more oxygen released, the more photosynthesis is going on. How do these two graphs relate to one another? Michael?
Well, each of the chlorophyll and the carotenoids, how do you say it, those are affecting the rate of photosynthesis. So like when they're absorbing most light, do the most photosynthesis. And he also noted that the carotenoids, how do you say it, it's actually not really affecting photosynthesis too much because it peaks back up near the end, where there is none, it's not absorbing any light. Okay. And then the chlorophyll A also seems to be the best at photosynthesis, or the most common in both of them.
Okay, yeah, absolutely. That's interesting what you noted about the carotenoids here, and that it peaks up almost as much as it peaks up here, so therefore it must not be doing a whole lot for photosynthesis, so that's interesting. We're going to learn tonight I'm going to talk in the video a little bit about carotenoids. One of the main reasons for these is they absorb wavelengths of light, like this part right here, this higher energy that could be damaging to other pigments. And so that's one of the main focuses on that. Notice here, very little photosynthesis in the green-yellow range. Why does that make sense? Because what's happening in the green-yellow range? Most of the light is being reflected, right? And so you would expect that there would not be much photosynthesis there, all right? So therefore, if we were to answer down at the bottom, the first question, which wavelengths of light are most effective in driving photosynthesis, we would say in the 400 to 500 range, or violet to blue light, and about 650 to 700 range, which is more red light, okay? All right, and so, very good. The packet that you got, the staple packet that says photosynthesis, I wrote here photosynthesis review packet 1 through 9. That's based upon what we've talked about already. This is going to be, I'm going to use that packet as your review packet for our next test. So I just put 1 through 9 as that's what we've covered so far. But as we go through and work in class, you'll have time to work in class on it. So it's not going to be due tomorrow. All right, 1 through 9, I'm not going to check that you did it. But it'll just be due when you come in to take the test. Okay? All right, so it'll be an ongoing project. All right? But the video is for